take their seats. My name is Franck Polleux. I co-organized this symposium, but I'm going to start these concluding remarks by giving credit where credit is due, because as you probably noticed already, Francesca did most of the work organizing this meeting. It was her original idea, and I just tagged along and helped whenever I could, but I really didn't do much. Francesca really deserves most of the credit, so we should thank Francesca for organizing this. I'm going to just give a couple of thoughts on, as concluding remarks, and say, first of all, thanks to all the speakers who I thought gave fantastic talks. Thanks for coming. Thanks for sharing your enthusiasm and sharing all these exciting results. I think it's probably obvious to this audience that cell biology is, I think, the missing link in understanding normal physiological functions of neurons and non-neuronal cells in the brain, but it's especially relevant to understand disease states. It seems particularly relevant to neurodegeneration. I think what I was most impressed by all the speakers today is how multidisciplinary the approaches have become over the years. We've heard a lot of really fabulous applications of cell biology, cell biological tools, including fancy imaging, but we've also seen a lot of other techniques applied to understand the disease mechanisms underlying neurodegeneration. So I think, to me, even in a wider context, cell biology is really the missing link between understanding what genes are doing with actual finding causality to any sort of cellular pathways. And so I think the future is bright, and there are clearly a couple of areas, not only microtubule and cytoskeletal dynamics, but things like organelle function that seems to be really exciting new areas to pursue. And that leads me to introduce today's keynote speaker. We invited Pietro di Camilli to give this keynote lecture. So I have the privilege of introducing Pietro. So Pietro comes from Italy, as you know. He got his MD degree at University of Milan, and then he did a postdoc with Paul Greengard when Paul was at Yale, and Pietro became an assistant professor right away after his postdoc, and then he went back and forth a few times between Milan and Yale, but then since the mid-80s, basically, he's been at Yale ever since. He's now a John Klingenstein Professor of Neuroscience and Cell Biology at Yale. He's been the chair of Cell Biology for a while, and he's now actually the chair of the Department of Neuroscience. He received many awards. He's a Howard Hughes Medical Institute investigator since '92. He's a member of the National Academy of Science and Medicine, and received many awards over the years. I would say Pietro is a giant in the field. He has made incredibly numerous and significant contributions to the field of neural cell biology in general. I think he's been most recognized for identifying some key functions for phospholinositides at synapses. Pietro has spent most of his career on the presynaptic side, where he's really transformed the field, not only looking at phospholinositide functions and lipid dynamics in general, but also dissecting in a very mechanistic way the functions of new and proteins of unknown functions presynaptically, including key regulators of presynaptic recycling, such as dynamines, but also characterizing many other proteins presynaptically, from phospholipid kinases to phosphatases, synaptogenin, and over the years, he also ventured into new territories, defining classes of proteins based on structure-function relationships. I'm thinking specifically about the bar superfamily of proteins, including really elegant characterization of proteins containing F-bar domains, which are both curvature sensing and curvature-inducing, membrane curvature-inducing proteins, and made really fabulous contributions in that field too. I think I'll finish on a couple of personal notes, if I may. The first one is that, in my opinion, Pietro is scientifically a renaissance man. If you think about, and I think he'll illustrate that beautifully in his talk today, Pietro uses a wide array of approaches to tackle, in a very mechanistic way, the function of all the proteins I mentioned before, presynaptically, from mouse genetics, knocking out multiple, making triple knockouts, for an entire family of proteins, CREM, electrophysiology, of course, cell biological approaches, biochemistry. It's always very impressive to read Pietro's papers, and humbling in many ways. And I have to admit that, I don't know if Pietro realizes this, but in 2003, I believe, so almost 15 years ago, he organized a meeting in a small place in France, by a foundation called Letray. He co-organized that meeting with Yukiko Goda, I believe. And I was fortunate enough to be invited there. I still don't know why, but I benefited tremendously from this meeting. In fact, I have to admit that I'm sort of a cell biologist today because of that meeting. I was profoundly influenced by that meeting, by the quality of the science that was presented there. And so I'm grateful for Pietro for this and many other things. So I'd like to give a warm welcome to Pietro on stage. Thank you.
Thank you, Frank, for this wonderful introduction. And thank you to the organizer, you and Francesca, for organizing this wonderful meeting. And I have to say, you said good thing about my work. I would like to say that this has been possible because of many wonderful collaborators that I had in the lab, some of whom are here in this room. So my lab has been interested. Can you hear me? Yeah. My lab has been interested throughout my career in the molecular mechanisms that control membrane dynamics and membrane traffic. And in particular, I have extensively used for the studies the neuronal synapse, which is a compartment highly specialized for membrane traffic. And this is illustrated in an electromicrograph and in a cartoon in these slides here. The nerve terminal is, obviously, its main specialization is to mediate the neurotransmitter release via diffusion of synaptic vesicle, which then reform very rapidly by endocytosis. This pathway of recycling is highly interconnected with other recycling pathways. They control housekeeping, clathrin-mediated endocytosis, recycling receptor or transporter, etc. And is also interconnected with membrane traffic to and from the cell body. Obviously, in our work in this area, we have focused on a lot of different proteins. But what really was seminal in our work and what led me to the topic that we'll discuss today is our identification of the important role of lipids of the bilayer. Obviously, the lipids of the bilayer are the key constituents of the membranes, but we have found that some proteins interact directly with the bilayer, some proteins function as curvature sensors, curvature generators, but in particular, that the chemistry of the bilayer is very important in controlling the vectoriality of membrane through these various pathways, and the metabolic changes that occur on the surface of the membrane as membrane move from one compartment to another are critical to control this traffic by inducing the shedding and the recruitment or the activation of specific factor. So this in turn led us to become interested in the mechanisms that control membrane lipid homeostasis. And this is the topic that I will discuss today, and actually we'll move away for today from membrane traffic and will describe other important functions of membrane which lead with their close proximity, but not with the membrane interaction or membrane fusion and fission as typically occur in membrane traffic. So I say that I'm interested in mechanisms to control lipid homeostasis, and one major mechanism to control lipid homeostasis has recently been shown by many studies, the direct transport to lipid and membrane contact site, contact that would lead directly to fusion. And in my talk today, I will illustrate first some example of membrane contact site, and second, I will briefly mention some protein that have been investigated primarily the air to plasma membrane contacts that mediate this lipid transport. And this will be to introduce the main topic of today's talk, which is the role of a family of proteins that function at intracellular contact site between the R and either the mitochondria or lysosome, which are important in neurodegeneration because their mutation lead to severe neurodegenerative diseases. So first, a few words about the introduction to membrane contact site. Most bilayer lipids are synthesized in the endoplasmic reticulum, and from there, they are transported to other membrane by classical membrane traffic. Vesicle which fuse with the Golgi, and then new vesicle emerge from the Golgi, and they reach different destinations. However, this pathway of delivery of newly synthesized lipids, and actually the reverse pathway for the return to the ER of metabolic product lipid for membrane recycling, can only occur in a relatively slow fashion, while some lipid turn over extremely rapidly. In addition, this flow, if it was not compensated by other mechanisms, would slowly result in randomization of lipids. Instead, there should be mechanisms to control specificity of the lipid composition of different membranes. And finally, this pathway would be inadequate to deliver lipid to mitochondria, which are not connected to the endoplasmic reticulum by membrane traffic. So what the work of many different labs over the years, including my own, has shown is that there are a major mechanism to control the delivery of lipids to other organelles and exchange of lipids are lipid transport protein, and which shield the lipids in an hydrophobic pocket, and they extract it from one membrane and deliver to another membrane. And so basically the way to allow this lipid to transfer the aqueous environment of the cell independent from the cellular traffic. The occurrence of this protein had been known since the 1970s, but it's only recently it has become clear that most of this protein act at site where two membranes are in closer position. And this is possible because endoplasmic reticulum has contact with virtually all organelles of the cell, as shown here, with lipid droplets, peroxisome, lysosome, Golgi complex, mitochondria, plasma membrane, endosome, etc. Two examples of such contact are shown here by cryo-EM. Here you see contact between the ER and the mitochondria, done by my colleague John Yu at Yale. And you can see here, this is a ribosome, so this is clearly the ER. You can see here the internal membrane of mitochondria, and you see here a region where the two membranes are closely opposed. You see that there are some densities there, and this is a collection of protein with many different functions, but one of the functions is to transport lipids. Here is a contact between the ER and the lysosome, and you see here this very close position. And now I want to give you a three-dimensional view of these contacts and also their abundance in neurons, which are our favorite cells. To do this, we use focused ion beam scanning electron microscopy, which is a technique which allows to analyze by EM large volumes of brain tissues, or any tissue for that matter, prior to this technique, at the most one could examine only hundreds of nanometers using tomography, using electron tomography, but focus ion beam really allows you to analyze much larger volume, and the idea is to use, instead of transmission electron microscopy, to use scanning electron microscopy. One take an image, and then one mill a little bit of the surface of the block while the focused ion beam, then another picture is taken, and so forth and so on. And then all these images are superimposed to generate movies, such as the one shown here, which allow to see with impressive resolution. Remember, this is a scanning electron microscopy, it's not electron microscopy. It allows you to see in a three-dimensional way, the internal structure. And notice, focus, for example, this endoplasmic reticulum with ribosomes. The endoplasmic reticulum made very close contact with the plasma membrane. You see here a big density because there is almost no lumen there. The two faces of the cisterna are tightly opposed. But if you see when I focus up and down, you see that it opens up at the other side. So this is really contact between the ER and the plasma membrane. There are contact between the ER and mitochondria, ER and lysosome, etc. Then one can take this movie, as you may view in my lab has done, and it has generated three-dimensional reconstruction shown here. And you can see the endoplasm. This is a neuron in the striatum. The yellow is endoplasmic reticulum, the green are mitochondria, light blue are endosome, magenta are the synaptic site at the cell surface. And you can see how these various organelles are interconnected to each other. And in red, for example, are the contact between the ER and the plasma membrane. These are sites where the cisterna of the ER are clustered at the plasma membrane, connected by small molecule where many things happen, includes lipid transport. And these are contact between the ER and mitochondria, for example. In the cell body, about 10% of the plasma membrane is covered by this contact. In the dendrites and axon, as you will see in a minute, much less. Mitochondria is about 4 to 5%. These are contact between the ER and the mitochondria. This is a dendrite. And you can see, again, the endoplasmic reticulum really penetrates every region of the dendrites. It ends up in dendritic spines. And in purple, in magenta are the synaptic site. And you see here the contact with the plasma membrane in red. They are smaller than the cell body, but they are there. The contact with mitochondria are here. And notice, by the way, in connection to the talk we had earlier this afternoon, how elongated mitochondria can be in certain dendrites. And they establish beautiful contact with mitochondria. And finally, with AR. And finally, the axon and axon terminal. This is the reconstruction of an axon. And you can see the endoplasmic reticulum is a single element of the endoplasmic reticulum that penetrates the entire axon from beginning to end. In larger axon, obviously, there are more tubules of ER, anastomose tubule, but in the small ones, it's just a single one, and then open up and branches in an terminal, and then continues, and then open up again, and then continues. And these are synapses with cluster of synaptic vesicle, and this is shown at better magnification here. One of these nerve terminal. You see the cluster of synaptic vesicle, the synaptic junction in magenta, and you see the ER, contact with the plasma membrane, and contact with the mitochondria. And so we are interested in the lipid exchanges that take place at these sites. So these contacts, which do not lead to fusion, have many functions. Control of ion fluxes. In many cases, these are sites where ion are moved from one membrane, from one organelle to another, from the outside to the inside of the cell, or for example, from the ER to mitochondria. There are places where some protein anchored to a membrane act in trans on another membrane, enzyme. For example, there are protein phosphatase in the ER that act in trans or endosome or on the plasma membrane. And relevant to the talk today are sites where lipid transport and or exchange with a bilayer fusion occur, and there are other functions as well. In connection to control of ion fluxes, I would like to remind that what we discussed this morning about awry and STIM1 is actually something that occur at this contact site. This contact site, all of them, no matter which organelle they involve, are highly dynamic structure which are populated by different components at different moments, depending on the functional state of the cell. The idea is that molecules move very rapidly in the DR. In a fibroblastic cell, a given molecule visits the entire DR in about 10 minutes, as shown originally by beautiful work by Jennifer Lippico-Schwartz. And so the idea is then when the protein migrates through the DR and encounter a contact, if the conditions are favorable to establish the contact, the protein will get stuck and immobilized there, as shown here by these blue colors. And then the condition of this in the cell change, for example, phosphorylation, increase of calcium, etc. The protein can be shed, and new protein can be recruited. So these are very dynamic structures. So again, I say lipid transport and exchange is what interests us today. We have worked on several such proteins over the last few years. So for example, one class of protein we have extensively investigated at the R to plasma membrane contact site and also at contact between the R and other organelles are the ORP family proteins. These are proteins that contain the so-called ORD domain that can contain more than one phospholipid. One of them typically is PI4P, and the other one can be cholesterol or phosphatidylserine or other. They are anchored in the ER either directly, as in the case of two members of the family, or 5 and ORP8, or indirectly through VAP, a protein that binds many proteins with a small sequence called FFT motif. And at this contact, this protein mediate counters transport. They bind in trans PI4P on another membrane, can be the plasma membrane or endosome or the Golgi, and they counter transport on the other direction another lipid, cholesterol, phosphatidylserine. And for example, we have shown that these proteins here are critical to enrich the plasma membrane in phosphatidylserine. They transport PI4P to the ER, where PI4P is degraded by a phosphatase, and this allows the phosphatidylserine to be brought to the plasma membrane. This model was first developed for one of the members of this family by Bruno Anthony, but it will now appear. It appears now that's a very general model. If you knock this protein down, there is a dramatic ectopic accumulation of PI4P that lead to dramatic cellular defect. So it's very important for cell to have this type of contact in order to, as I said at the beginning, to homeostatically control the lipid composition. Another class of proteins we have investigated are proteins that contain SMP domain. And in particular, we have worked extensively with the two family of proteins, the extended synaptotagmin and C2CD2 and its paralog C2CD2-like, also called TMEM24. These are proteins that are anchored in the endoplasmic reticulum via either hairpin, in the case of extended synaptotagmin, or by transmembrane region, in the case of C2CD2 and C2CD2-like. And they reach the plasma membrane in trans via binding site, which are mediated by the extended synaptotagmin, for the extended synaptotagmin by C2 domain. Each of these red circle is a C2 domain. That many of you know that there are phospholipid binding domain, often also sensitive to calcium. And in the case of C2CD2 and its paralog, by positive charges at the end of the molecule at the C-terminus. By the way, notice that both of these molecules are dimeric molecules. And they dimerize via the SMP domain, and this SMP domain, as you will see in a second, they are lipid transport module. These two proteins here mediate rapid homeostatic adaptation in response to stimulation. So they are not permanent member of contact site, but they associate and dissociate very dynamically so that the function of lipid transport is highly regulated. And lipid transport is regulated by this SMP domain. So I will show you, we have extensively studied this protein. I will show you just 3 slides to convince you that this regulated lipid transport protein. So first of all, I show you they have a very dynamic behavior. The extended synaptotagmin, which, by the way, I want to emphasize, they have this name of synaptotagmin because they are very reminiscent, but very remotely, of the classical synaptotagmin, the mediate tethering between synaptic vesicle and the plasma membrane to mediate calcium-dependent fusion. but these proteins do not lead to fusion. These proteins simply mediate calcium-dependent tethering. What you see here is a fibroblast in which we knocked in at the endogenous locus of extended synaptotagmin 1 and EGFP, so that now you see extended synaptotagmin at its normal level of expression, and what you see here is an ER localization. But if we increase calcium, there is a very rapid clearance of the protein from the bulk of the ER, and now it's accumulated at the site of contact. And this happened because some C2 domain of extended synaptotagmin are exquisitively calcium sensitive and therefore induce this accumulation at the cell surface only when calcium is elevated. Conversely, this is TMEM24, also called C2-CD2-like, in neuron, because I should mention that the extended synaptotagmin are not present at high concentration in neuron, are only present at high concentration in other cell types, for example, the immune system. Instead, TMEM24 is highly expressed in neuroendocrine cells such as pancreatic beta cell and in neuron. This is a neuron in culture. These spots here represent contact sites. By the way, these contact sites are also enriched in a variety of ion channels such as Kv2.1 and Kv2.2 channels and in the L-type calcium channel. And when calcium is elevated, and we can do that by NMDA activation, stimulation, by electrical stimulation, or by depolarization with potassium, you can see that the protein dissociates. It's exactly the opposite behavior. In this case, it's mediated by the phosphorylation of this site here. And you can see here the protein dissociates in response to depolarization, becomes distributed to the air, and then is recovered when we remove the potassium chloride. So this is just to show they are dynamic protein. This to show you evidence that they are really lipid transporter. In collaboration with Karin Reinisch, my colleague at Yale, we determined the crystal structure of the SMP domain of extended synaptotagmin and subsequently also of TMEM24. And as you can see, is a dimeric structure. This is one monomer, it is the other monomer, that contain some lipids. And by mass spectrometry, by isolating the SMP domain from mammalian cell, we have found that the protein can contain, without real specificity, glycerophospholipid. And this is a different representation of the molecule. These are the lipids, and it shows that there is a deep hydrophobic cavity. So light gray is hydrophilic, surface exposed. Dark blue is hydrophobic, and this is a deep groove within which there are these lipids. This is the acylglycerol chain of a phospholipid. This is triton because the crystal structure was done using the protein purified with triton, but we know that in cell we have only glycerolipid. This module can transport lipids, and this is an in vitro experiment that clearly proved the point. Here we've used liposome, a septor endonal liposome, and we have used a Fred-based assay to measure the transport of liposome of fluorescent glycerolipid from one liposome to the other, and we have found that this transport does not occur if you don't put any protein. If you put the protein without calcium, there is a minimal degree of transport, but if you put in the presence of calcium, there is a very dramatic transfer of lipids. And we know that this is not due to fusion, because at the end of the reaction, we can still separate the liposome by adding proteases and a DTA. And also we have found something very interesting. Xinbian in my lab has found that this calcium dependence is due to two factors. First, because the protein only tethered the ER to the plasma membrane, in this case, tethered to endosome only in the presence of calcium. And second, because one of the C2 domain is bound in an autoinhibitory conformation. So basically, in low calcium, occlude the opening of the lipid channel of the SMP domain. And when calcium is bound, this autoinhibitory interaction is lost, and then the protein can transport lipids. You have extensively worked on trying to understand the physiological function of this protein. We have generated mice cells. It turned out that it's not it's not easy to study physiological function. There is a lot of redundancy of lipid transport proteins. So the defects are very modest. Nevertheless, some modest can be seen. Here, one example of this defect. We have taken these HeLa cells in which Yasunori Sakai did a triple knockout of the 3 extended synaptotagmin, and now we have a triple knockout of mice, which, by the way, do not have obvious defect, but now is more intense analysis that seem to be defect in particular in the immune system. Anyway, when you stimulate a cell, for example, by activating with an agonist the cleavage of biphospholipase C of PIP2, there is the generation of diacylglycerol and IP3. Then diacylglycerol need to be recycled. This is an hydrophobic molecule that remains in the membrane, partially is transformed in phosphatidic acid, but then both DAG and phosphatidic acid need to be brought back to the endoplasmic reticulum for metabolic recycling. And we need some transporter to do so, and extended synaptotagmin play an important role in this transport, at least for DAG, as we have shown by TURF, which is a microscopy technique that allows to measure the accumulation of fluorescent proteins selectively at the cell surface, where we lose a diacylglycerol sensor, and which is shown here, and this is the response to an agent that activates phospholipase C, ionomycin, and you can see that there is a very rapid increase of diacylglycerol in wild-type cell, which are in blue. In the absence of the extended synaptotagmin, the recovery is much delayed, and this recovery is brought back to more normal rescue by reintroducing extended synaptotagmin 1 and 2, the form a dimer, and this is a quantification of the result. So this is just to tell you that this SMP domain are lipid transport module present in some protein present at contact site. But now this leads me to what I really want to talk to you about and relevant to neurodegeneration. So the SMP domains are a group of domains which are members of a large superfamily of protein, which are the tulip domain. And within this large tulip domain family, one can identify different branches. And this is from a review by Alba and Lupas, a bioinformaticist with a bioinformatic group that work at the Max Planck in Germany, where each of these spots represent the SMP domain of a protein, and you have so many spots for each protein because they bioinformatically predicted the structure in different species. So this is a different protein, but also different species. The distance between all these spots indicate the predicted structural difference between these various domains. So the SMP domain are one group of these tulip domain which act intracellular. And one class are the one present in the extended synaptotagmin, and the other one are the one present in TMEM24 and C2CD2. All these module here, all these SMP domain, are present in extracellular proteins. For example, CETP, which is cholesterol ester transfer protein that circulates in the blood, and these are other also protein, lysophospholipid binding protein, etc., all protein that acts extracellularly. But interestingly, are all structurally related. This is the structure of the SMP domain of extended synaptotagmin and C2CD2 or TMEM24, as determined in collaboration with Karin Reinisch, and this is structure present in the literature of this protein that work extracellularly. So really, we are dealing with a family of proteins specialized for the transport of lipids, and nature has evolved in a slightly different way, the protein that work outside or inside, but using essentially the same basic model. If we focus on the SMP domains family, the one that work intracellular, you can see that there are some other ones that I didn't mention. One, for example, is the PDZ domain containing protein, also called PDZ-D8, studied by Franck Polleux. And then there are M1, MM1, MDM12, and D34, which are very interesting because are part of a complex of 4 proteins. So these are three proteins within a 4-protein complex known to act at the boundary between the endoplasmic reticulum and mitochondria. So let me tell you 2 words about this contact. You have already seen this image, a contact between the mitochondria and the ER, and also this image, which is the contact of mitochondria and the ER in an N-terminal. These contacts are extremely important because the mitochondria are highly dependent on the endoplasmic reticulum for the makeup of lipids. So they can modify and also make some lipid, but they use a lot of precursors that come from the endoplasmic reticulum. And conversely, they modify some lipid provided to them by the endoplasmic reticulum. They send them back to the endoplasmic reticulum. And some of this arrow indicated this exchange. This is endoplasmic reticulum. For example, PA goes to the mitochondria where it's used to make cardiolipin. Phosphatidylserine is transported to the endoplasmic, to the mitochondria. They make PE out of that, phosphatidylethanolamine, that they come back for generating phosphatidylcholine and also to provide to the cell with phosphatidylserine. So this contact, this exchange of lipid is very important. But so far, we knew very little about which protein mediates this exchange. So when Peter Walter identified this Hermes complex, the idea was that perhaps the problem was solved because the Hermes complex contained 3 proteins which have SMP domain, and now we know the crystal structure of some of these, actually of all of this domain, no, I think 2 of this domain, and they are extremely similar to the crystal structure of the SMP domain of stendesinatotagmin 2. This was the founding member in thermocrystallography. There is one problem, however, that the Hermes complex is not conserved outside the fungi. It's not present in some unicellular organisms outside the fungi and is not present in the metazoa. So the question is, since also in metazoa, the mitochondria and the ER are interdependent for lipids, how can this happen if we don't have an Hermes complex? So there is at least one protein. So before I go on, I want to say the following, that however, it was found that in yeast, if you don't have the Hermes complex, the cells are sick, but they can live, suggesting that there is something else. And so the idea is maybe in mammal, this something else has become important. I should mention that there is at least one protein, PDC-D8, which is related to MMM1 in mammals that Franck has shown to be reported to be a contact between AR and mitochondria. However, there is no evidence of a complex like the Hermes. So it was very important to understand what this other potential compensatory pathway was. So work in yeast, so this is a cartoon summarizing what I told you so far. The Hermes in yeast mediates lipid transport. 2 independent groups in 2014 reported that while yeast cells can live without Hermes, they cannot live if in addition to Hermes, another protein complex is deleted, of which a key component is the protein BPR39. And this investigator has shown that this BPR39 is localized at contact between the mitochondrion and the vacuole, which is the yeast lysosome, suggesting there may be an alternative pathway to deliver lipid via the vacuole. The problem, however, is none of the components of this complex, called V-clamp, had any lipid binding domain, any at least putative lipid binding domain. But things became more interesting when 2 groups, the first one was Corman, reported that Hermes deficiency, so the sickness of yeast cells that don't have Hermes, can be bypassed by dominant mutation in another protein called VPS13. And interestingly, they found that VPS13 was present at contact between the vacuole and mitochondria, or the vacuole and ER, depending on the growth condition, confirming and supporting this idea that there is an alternative pathway to deliver lipid to mitochondria independently of the Hermes complex. VPS13 is a humongous protein, one of the 5th largest protein in yeast, with a domain structure that until recently was not known, with the exception of a known pH domain at the C-terminus and the putative WD40 motif right here. It had been previously studied in yeast prior to the finding that a dominant mutation suppresses lack of Hermes. It has been identified in a variety of screens in a variety of contexts. It had been shown to be involved somehow in correct trans-Golgi network to endosome transport. Was shown to be needed for the growth and closure of the sporulation membrane or yeast. And keep this in mind, I will come back to that. And it was also shown to be important to maintain mitochondrial integrity. And again, I will come back to this concept of mitochondrial integrity at the end of the talk. Another interesting thing about this protein, it had been noted that this protein has a regional similarity to a key autophagic factor, ATG2, which is important for the growth and the closure of the autophagic membrane. And in particular, there were sequence of primary amino acid similarity, region of primary sequence similarity at the N-terminus and the C-terminus that have been called ATG homology region. Now, this protein came to our attention for two reasons. First, because we were interested in SMP domains, and there was this SMP domain protein at the contact between the ER and the mitochondria, and their function seemed to be suppressed by mutation in this protein, so we thought that this could be a lipid transport protein. But also, as neurobiologists, this protein came to our attention because mutation in this protein, and there are 4 genes encoding VPA13 in mammals, each of them is responsible for neurological neurodegenerative diseases. VPA13A, also called choline, is responsible for chorea acanthocytosis, which is a Huntington-like syndrome, which in addition, patients with this syndrome have defect in red cells, which acquire the shape of a leaf of acanthus, so they're called acanthocytosis. So this is VPA13A, and Huntington-like neurodegeneration. VPA13B is responsible for a developmental defect, they collectively called Cohen syndrome, which include, among other defects, neurological defects. VPA13C had been identified in a mutation in familiar form of early onset Parkinson's disease, and more recent work, partially unpublished by others, has reported that in fact VPA13 mutation are a risk factor in Parkinson, and there appears to be a really major protein in early onset Parkinson. Finally, VPA13D is mutation responsible with ataxia with spasticity or with childhood movement disorder. So all of this, and the fact that this protein may be a lipid transport protein, in addition to other things that I don't have the time to explain, brought this protein at our interest. And so we decided to focus on that, and again to collaborate with my structural biology colleague at Yale, Karin Reinisch. So we collaborated with a PhD student, Nikit Kumar, and in my lab, the key players were Mariana Leoncino, a wonderful postdoc, and Willan Conceruti, a wonderful MD PhD student. So first, is VPA13 a lipid transport protein? And this is where Karin became very critical as in help. She was able to purify from yeast, from the single yeast protein, this N-terminal region, and she found that when this region was incubated with fluorescent lipid, this lipid became part of the protein so that it could migrate together with the protein in native gels. And these are Comassi blue gel showing the protein, and you can see here, this is the fluorescence, and you can see that when the protein was loaded with PE or PS or PA, sterol and ceramide, at least with some lipid, this lipid migrates with the protein. So this tells that there is a cavity inside. We also performed a lipid transport assay, similar to the one I described for extended synaptotagmin, and we found that the protein can transport lipid, in this case, not in a calcium-dependent way, just constitutively. You can see here some controlled transport, and this in the presence of VPA13 alpha. We call alpha this domain here, and this is a controlled lipid transport in the presence of extended synaptotagmin 1. Now, the crystal structure. So far, we don't have a crystal structure of this whole region here, which seems to be the critical player in lipid transport. But Karin and Nikit were able to crystallize the N-terminal portion of this protein from a fungus, Chetomium thermophilus, which is often used by crystallographer because protein from this organism are more stable. And it turns out that this is a beautiful structure, which essentially like a hand, closed hand, in which the inside is completely hydrophobic. You can see here red and blue are hydrophilic, the positive and negative charges, and in gray are hydrophobic, so this is ideally suited to contain lipid. And actually, when she made a longer fragment and then yet a longer fragment, it found that the longer it was, the more lipid they could contain. And this is shown here. This is, for example, MBD phosphatidylethanolamine, MBD phosphatidylserine. The longer the fragment, the more lipid it was. And this led to the idea that this is very long, elongated fragment that contain multiple lipids. And in fact, the stoichiometry is remarkable. There seem to be as many as 20 lipids per molecule. So this proved that this protein has something to do with lipid transport and also with lipid binding and lipid transport. Now, if this is the case, where does the protein achieve its lipid transport function in mammals? So I told you that in yeast the protein was located at the contact between the vacuole of mitochondria, the vacuole, and the ER. So we started with, we focus in particular so far on VPA13A and VPA13C. VPA13A is the one responsible for chorea acanthocytosis, which I described earlier. And when Mariana expressed this VPA13A labeled with fluorescence, so this is an overexpression in cell, she found that it was beautifully localized at the interface between the ER and mitochondria, the place where in yeast we have the Hermes complex that does not exist in mammals. Green is the ER, blue is mitochondria, VPA13A is the protein, and you can see here at higher magnification the protein is only at places where the ER intersect mitochondria. And here also you can see this is a mitochondria, you see hotspot of VPA13, and these are the point where the ER intersect mitochondria. Obviously, could this be an artifact of overexpression? So they tag an HA tag, they put an HA tag at the endogenous locus, and then they use immunofluorescence to visualize this HA tag VPA13. In this case, the contact are not so extended, they are hotspot, but the majority of spots, they are localized on mitochondria. You will see that, as you can see, there are some spots not on mitochondria, and we understood this cell, what the spots are when you look more carefully, and it turns out that in cells where there are lipid droplets, then you have VPA13A, also lipid droplets. So not only at contact between AR and mitochondria, but also around lipid droplets. And actually, the overexpression of VPA13 induced a complete wrapping of lipid droplets by ER. And you can see here at high magnification. So it functions as a tether, so it tethered ER to mitochondria, but also the ER to lipid droplets, and it completely induced the enraption of lipid droplets by the ER. And you can see here correlative EM, fluorescent, and EM. You can see VPA13 and the ER. And you can see if you overexpress the protein, it induces a dramatic expansion of this contact. So how does VPA13A tether organelles? And by the way, as you will see, what we have done so far with this protein has been done in non-neuronal cell, but now there is a lot of work going on as we speak to attempt to understand this protein in neuronal cell, where they have the major phenotype. How does the VPA13A tether organelles? Well, we found that the N-terminal region bind to the ER and the C-terminal region bind to mitochondrial lipid droplets. So the N-terminal region contains an FFT motif, which you may recall I said is a binding site for the ER protein VAP, which is shown here. And in fact, if you express only this fragment in cell, you have a beautiful localization with the ER. If you mutate this FFT motif, the protein becomes diffuse. If you express the C-terminal region, this fragment goes to mitochondria. You can see the surface of mitochondria and lipid droplets. Interesting, here there is a region which I remember I told you has homology to a TG2, the so-called TG2 homology domain. The TG2 homology domain of a TG2, which is the autophagic protein, was shown to go to lipid droplets, And this, in fact, would drive VPF13 to lipid droplets. And if you mutate an amphipathic helix here, you no longer have this localization. So about VPF13A. VPF13A is a protein located at contact between ER and mitochondria, ER and lipid droplets. We think is a conduit for the movement of phospholipid between these various locations. And clearly, we believe it's very important to maintain appropriate traffic of lipid between ER and mitochondria. Notice that in yeast, the protein was not a contact between ER and mitochondria, and instead was a different location. So during evolution, this protein is split in 4 genes. The gene is split when there went 2 duplication, and at least one of them had acquired what was originally the function of the Hermes. What about VPF13C? A and C are the more closely related member of the four VPF13 in mammals. VPF13C was identified as a gene mutated in Parkinson's disease. It's a little bit longer because there is a duplication of this region here, which we think we make this lipid binding pocket even longer. The investigator that studied, that reported, did the first report of VPF13 in mutation in Parkinson, reported the occurrence of some mitochondrial defect in cell without VPF13C. So having seen what you've just saw, having done the experiment that I just showed you, we were very excited to say, okay, we have another protein also localized at the R2 mitochondrial contact site, and that will explain what this investigator had reported. You can see the title of this paper. To our surprise, VPF13C had a very different localization in cell than VPF13A. Here is A, which is located in mitochondria, and C, which is different location. And we found that this location were late endosome and lysosome, in addition also to some lipid droplets. And this can be seen here. If you overexpress the protein induces a complete enwrapping of late endosome and lysosome by the endoplasmic reticulum, and is there where the protein is localized. And again, when we did the endogenous tagging, most of the spot for VPF13C are located at interfaces with the late endosome. They happen to be RAP7 here, but lysosome will be the same. And some spots which are not colocalized most likely represent lipid droplets, because the protein also binds lipid droplets. So how does the protein cross-link these various organelles? Again, the N-terminal region bind VAP, and the C-terminal region bind lipid droplets. You can see here this region. It doesn't bind mitochondria, and that was really very surprising. And the binding to endosome lysosome is mediated by the WD40 motif, and this is a fibroblast, but the same thing occurs in neuron. These are neuron overexpressing this fragment of VPF13C. So there were also independent evidence for a role, because we were so surprised by the lack of presence of mitochondria. We further investigated the lysosome, and you can see that in VPF13C knockout cell, there is an increase in LAMP1, which suggests that this protein really has something to do with lysosome. As you also probably know, an important protein that signals at the surface of lysosome is TFAB, which is a protein that translocates from the surface of lysosome to the nucleus when there is a need to make more lysosome. And we found that, in fact, in VPF13C, more TFAB was localized in the nucleus than in control cell. So all of this corroborates the idea that this protein is something to do important with lysosome. So this is a summary of what I just showed. These two proteins have similar yet somewhat different localization, different type of contacts, but the idea is, again, that they mediate this flux of lipid between these various membranes to maintain lipid homeostasis. Again, going back to the yeast protein, in yeast, the single VPF13 was localized at interfaces between the vacuole and mitochondria and the ER. It's interesting we found here a protein localized at interfaces between the lysosome, which are the equivalent to the vacuole, and the ER. And we think that maybe as the gene duplicated, different genes acquired different properties of the original VPF13 in yeast and fungi. What about VPA13B and VPA13D, the other two? We know that VPA13B also goes to mitochondria, but we don't have other information so far. VPA13D, however, we haven't studied, but it's been reported to be localized in lysosomes. And in fact, there are a couple of papers that came up last year showing that impairment of VPA13D function results in defective mitophagy and increase in mitochondrial size. However, at least one of these papers tells that the protein is on lysosomes, so that is an indirect effect which is attributed to a lack of mitophagy. VPA13D contains a domain that binds ubiquitin, in particular a K63 ubiquitin chain, so it's thought to be an adapter to bring the mitochondria to the autophagic system. An interesting lack of VPA13D is phenocopy by lack of 2S-core component. VPA13D was identified in a screen for defective mitophagy in intestinal cell, which also led to the identification of this protein here, so strongly suggests an interplay between its function, the autophagy system, and mitophagy. So, as I said, we were surprised by the fact that VPA13C, which was linked to the Parkin-Pink pathway by the paper that published the first study, that was not on mitochondria but on lysosome, late endosome. But we are struck by the fact that if you look at the best characterized PARK gene, you see this recurrent motif of either endolysosome or mitochondria, suggesting that there is a very intimate relationship. And today we had many examples of talks which mentioned the fact that damaged mitochondria are eaten up by the autophagic system, by mitophagy. And so one can easily understand how mutating one side or the other of this partnership, we may come up with similar manifestation. But now I would like to conclude by coming back to two points that I anticipated. The first thing I said, one thing I said when I first introduced VPA13, I said that VPA13 was identified in screen for mitochondrial integrity. And what do I mean by mitochondrial integrity? Mitochondria contain the DNA, and if the membrane of the mitochondria lose their integrity, some DNA can escape and end up in the nucleus. And so a screen has been designed in several years ago in which people look for genes that protect mitochondria from DNA escape. So the idea was they knocked out an essential gene yeast in the nucleus and they put in the mitochondrial genome. And if the yeast cell that would survive would be the one that are able to leak the DNA from the mitochondria and to go in the nucleus. And this screen led to the isolation of 6 genes. Probably there are more, but this gene reported 6. The so-called YME gene, yeast mitochondrial escape. Well, it turned out that YME4 and YME6 are components of the Hermes complex, and YME3 is VPA13. So this suggests that if you lose this yeast mitochondrial, this protein, probably we would say now, because of the defect in lipid, you lose some mitochondrial integrity with escape of DNA. Now, importantly, leakage of DNA from mitochondria triggers the innate immune response. So, as you know, the mitochondria are derived from original pathogens. You are well familiar with the endosymbiotic origin of mitochondria. And so cells have developed an innate immune response to defend them from DNA from this pathogen. And so when mitochondria leak DNA, they activate a response that has been developed to protect from pathogen. And it has been extensively shown and investigated that leak of DNA on RNA, of oxidized DNA, of other components of mitochondria can activate this innate immune response, which in turn lead to inflammation. And I don't need to tell in this room that inflammation has been implicated in Parkinson's disease, and for example, David Salzer is part of a very nice study on that. And so the idea is that if you lose mitochondrial integrity and DNA leak out, and we know that they can leak out because of the study, we can activate this innate immune response. And this obviously is a pie in the sky as an addition, and one new hypothesis that you can have Parkinson, but there is actually a paper that tells us that this can be the case. And I think many of you are familiar with this work of Richard Hughes that showed that mice which lack Parkin and PINK1 do not have obvious defect, but if they are stressed by exhaustive exercise, they undergo neurodegeneration. And they found that this results in the activation of the sting-induced inflammation, and knockout mice that don't have sting do not have this inflammation. And sting is really, going back to this image here, is the path, is part of the pathway that senses mitochondrial DNA. So I would like to propose that some of the neurodegenerative conditions due to the lack of VPA13 are due to this pathway. And finally, I want to come back to the second thing I mentioned early on that I say I will come back later, which is VPA13 and sporulation. So VPA13 was identified in screen for sporulation defect. So for those of you that are not familiar with yeast biology, when you have a yeast cell and the conditions are such that they are not favorable to growth, the cell eventually undergo two meiotic division and generate spores. And then the spores, basically each of these, this is a result of meiotic division, so these are really for yeast able to give rise to new cells when they were in the proper growth condition. During the formation of spores, so you had two meiotic divisions, so one nucleus becomes four nuclei, and before this happened, there is a new membrane generated at the spindle pole. This is really a cisterna that grows at the spindle pole and eventually surround the nascent nuclei and eventually closes, so that now during the spore, this will become the plasma membrane of the spore and everything else will be destroyed. But basically, we are dealing with the growth ex novo of a membrane. And if you don't have VPA13, the growth of the sporulation membrane is closed, is impaired, so leading to the possibility, and this is only a speculation, that VPA13 may deliver lipid in bulk because we know that it contains so many lipids. But there is something even more interesting, that the growth ex novo of a membrane is reminiscent of what happened in autophagy. And I told you earlier that there are small regions of sequence similarity at the N-terminus, the C-terminus of the protein ATG2, whose mutation are, whose absence lead to a strong defect in autophagy, and in particular to the growth and the closure of the autophagic membrane. So it's a process somehow reminiscent of this. So this is what I mentioned, this remote similarity. Here in the portion of the protein called, in VPA13, this region was called the corrin domain, and then there is a TG homology here. And a recent paper in Journal Cell Biology has shown that ATG2 can be present at contact site. So ATG9 established ATG2 dependent contact between the endoplasmic reticulum and phagophore. So, sorry, here, 2S here. Oops, sorry. So this really appeared to be a protein of contact site. So at Yale, we have a colleague, Tom Melia, which many of you know, that work on autophagy, and follow up this work we did with Karen Reinisch of VPA13, decided together with Karen Reinisch to study the structure of ATG2. And so this is their work, it's not my work, and I just brought a slide here of their work. They found that the N-terminal, this is a modeling, this one, the N-terminal portion of ATG2 can be modeled to be exactly like the crystal structure of VPA13, and in particular, the region of primary sequence similarity, the corrin domain, is what is here at the end, sort of the cap of this module. And then the module becomes just beta sheet and alpha helices, and probably all what is needed here is conservation of structure rather than precise amino acid sequence. And if you go on and you go on, in both VPA13 and ATG2, you have a continuous repetitive sequence suggesting that you can form an elongated structure here. But they even have a preliminary cryo-EM structure, a 15 extra resolution, that suggests, in fact, an elongated module with a cavity inside, and it is tempting to speculate that this cavity might be hydrophobic. And so this suggests the possibility that VPA13, like ATG2, may form a connection, possibly bridges, between membrane to deliver lipids. When I mentioned extended synaptotagmin and the OR protein in the beginning, I don't know if I emphasized that, but I wanted to mention that in that case, we think that this protein shuttle between a membrane and the other. So they are tethered at the two membranes, but the lipid transport module, we think, shuttle back and forth. In this case, the hypothesis that we propose that this protein might actually form a tunnel or a bridge to bring lipid from one membrane to the other. So to conclusion, and the conclusion will be of VPA13, join the family of lipid transport protein. Abnormal lipid transport may be responsible for diseases due to VPA13 mutations. VPA13 protein function as conduits for the transport of lipids between different organelles, including lipid droplets. Different VPA13 paralogs have different functions. VPA13A may account for the lack of the Hermet complex in metazoan cells. ATG2 is also likely to be a lipid transport protein. Obviously, there is much to do. We need to elucidate the further detail about the structure, mechanism of lipid transport, energetics. So is there an energy gradient? How would they move living around? What are the forces that mediate this transfer of lipids? Regulation. We need to better understand the physiological importance, distinct and overlapping function, because we know that if you knock out VPA13A or C, even in human, the patients are okay up to a certain point. We generated already A and C in mice, and there are also some paper reported. There is no major phenotype. We think that there is a great degree of overlapping, and obviously, in the case of neurodegenerative diseases, at some point this overlapping function may become insufficient, and that's why you have neurodegeneration. And obviously, we are very interested to understand mechanism of diseases. For example, VPA13A and VPA13C produce different diseases because they are primarily expressed in different cell type or because they mediate contact between different organs. We don't know yet. We only know that these proteins are ubiquitously expressed in brain. Maybe the ratio of the different ratio can vary, the relative ratio, but they are not selectively localizing some neuronal compartment. So, conclusion, interorganal communication at contact site is a very rapidly growing field. In the old days, and still now, if you look on textbook or on Google, you search cell, you find an image like this, where organelles are presented as individual entity. We know instead that the view of a cell from inside is profoundly different. All the organelles are tremendously interconnected. They talk to each other. These contacts have a variety of functions, including, obviously, as from my talk today, lipid transport. This inventory of contact site protein is far from complete. VPA13 is the most recent, sorry, I should say now ATG2 is the more recent member identified, the most recent lipid transport protein, the function at contact site identified. And so this field continues to be a fascinating area for new exploration in cell biology. And I would like to conclude. I have many wonderful members of the lab, but the work I reported today was done by Mariana Leoncino and Willianco Ceruti, which heavily collaborated with the Rheinisch lab, by they responsible for all the cell biology. Work on the SMP domain protein was done by investigators shown here. Some have left, some are still present in the lab, and the beautiful FIBSEM images I showed were done by Yumei Wu. I would like to acknowledge the critical collaboration with Karin Rheinisch, not only a great structural biologist, but a great biologist and a great colleague, and Harald Dessage and Ilya Farn for FIBSEM, and Jun Liu for cryo-EM. Thank you.
I guess I'll ask the first one, and then you so much.
Okay.
These lipid droplets are made and unmade so rapidly, and you have arrows going in a circle. Is this family responsible for their synthesis, degradation, or both?
Good question. I don't know yet. I know that actually VPA13C was also identified as a key component of adipose tissue, in particular brown adipose tissue. So we know that they have something to do with lipid droplets. Growth, I don't know. We don't know. Also, early days.
You didn't mention plasma membrane for any of the four members of this family.
No. So far, we have no evidence of VPA13 in plasma membrane. You're right.
I was wondering about the mutation for Parkinson's disease. So do you know exactly what the phenotype is in the late endosome lysosome when you have the mutation? What is the exact defect that's happening?
So far, we have those cells. So far, we haven't seen a defect. But, we just saw some increase in LAM2 and some increase in TFEB. But at the first observation, we don't see anything. Now, Will Anconcherut is busy to try to understand more. I think there is a large degree of functional overlap of this protein, of the various members of the family, yeah. And so we're obviously generating double and triple, yes, that's why.
So, Pietro, beautiful talk.
Thank you.
So, if EPS13A is part of something that's orthologous to the Hermes complex in metazoan cells.
I don't know orthologous, but clearly it's a different beast, but somehow
Sure.
participate, yeah.
Do you see any, do you think it's only involved in lipid transport, or do you have any evidence that it tethers? Do you see
the tethering.
By CREM, do you see changes in ER mitocontacts?
We haven't done the EM for the ER to mitochondrial contacts, but when we did the endogenous tagging or the overexpression, there was a very different length of the VPA13 positive segment, which strongly suggests that it also functioned as a tether. However, maybe your question relates to another more general question. Are there master tether at each of these contacts? And then these proteins are visited, they come and go. And my view, I'm a little bit agnostic on this. Perhaps there are many proteins that can function as tether, and it's a combination of this protein that create the contact, and in a given time, you take away some, you put back some other, but maybe there is not just a protein whose only function is to be a tether. Yeah.
Yeah. It's impressive to see the list of genes linked to diseases and neurodegenerative diseases. I wasn't aware of that. That's actually quite impressive. But maybe you did it by accident, or maybe there's a reason for it. I think FAB-B is an ALS gene.
Yeah.
How does it fit in your picture? So it's FAB-B, there's a specific mutation. Does it fit in your picture? Is
so
it also
VAP, as you know, we have investigated extensively. VAP is a ALS gene. And the only thing I can imagine is, VAP, you may say, well, it does create disease through VPA13 because it binds, but it binds so many other lipid metabolizing enzymes. I don't know.
So what exactly? What is the cascade? Is there a specific cascade there, or
in which way specific?
in FAB-B? So it binds to so many proteins. Is there in ALS or in neurodegenerative disease? Is it known which orb it binds? Or?
No.
Yeah, beautiful talk, Pietro. You've spent many years thinking about vesicular trafficking. I figured I'd ask a very broad question.
This is non-vesicular trafficking. The other side of the coin.
Right. So the question is, of course...
But these lipids are important for vesicular trafficking.
So, and there's also, of course, presumably cytosolic non-vesicular transport.
Yeah.
So can you speculate a little bit on the advantages of lipid transport as allowed by contact sites when compared to vesicular transport, or even cytosolic non-vesicular transport? What are the true advantages that you can think of?
One advantage is that, for example, the mitochondria, they are not connected by vesicular transport. But if you think of other things, it allows you to control the lipid composition of an organelle independently of how efficient or inefficient vesicular traffic is. Because you can accelerate or slow down a certain transport reaction, and if all your lipids were dependent on that, you will be stuck. So it makes sense to have two completely independent ways. So the question that you may ask is, could you do it simply by lipid transfer protein? But I think this adds an element of speed, but also an element of specificity. So that's why. So I'm struck by how many of the lipid transport proteins that have been identified in the 70s and later turn out to be tethered at membrane contact site. The first protein identified turned out to be near, which is binding site for the ER and binding site for the plasma membrane. Yeah.
Sorry, in the back. Hi, thank you. Beautiful talk.
Thank
I was
you.
just wondering, following up on that, do you think that there's any specificity for VPS 13 for lipid? And
It's
how
an
do you
interesting
envision-
question because the cavity is so big that it's very hard to believe how you can control specificity. Although we found, as you saw, that cholesterol doesn't bind cholesterol, but the glycerolipids seem to all go in, and also some ceramide. I'm puzzled, but it really looked like a humongous hydrophobic cavity.
And do you, how do you envision the polar head groups would get through such a hydrophobic region?
Well, but so the resolution of the cryo-EM is not good enough to make a conclusion, but I suspect that it might be like in the case of the tendency autotagmin SMP domain, that there is a seam. So it's not a tunnel, it's just a very deep groove. So protein can slide with the hydrophobic portion in the groove and the hydrophilic molecule sticking out in the cytosol. So that's a possibility. Yeah.
Gorgeous talk, Pietro.
Thank you.
This ATG2 result is really exciting. So I was wondering if you or your colleagues,
That is Karin and Tom Melia, yeah.
but it's in this greater picture, it's really
Yeah.
exciting. Have they shown that it binds lipid as well and have
Yes,
the same
yes.
high stoichiometry?
Yeah.
And does this slide that you're envisioning, is it like a slide like where there's a clear directionality, or is it possible it goes both ways?
Your touch, it goes back a little bit to
Yes,
the other
exactly.
question.
Yeah.
Because if you have a molecule at a time, you can imagine they can go one way or another. But if you have more than 10 molecules at a time, either you have a two-lane highway, or else can only be in one direction. And I don't know. We're trying not to think some in vitro system to really address mechanistically how that works.
Yeah, it's very
So,
exciting.
Yeah.
Okay. Well, I saw the late James Brown in Harlem, and they had to drag him off the stage. And I think Pietro's very similar. but we do have a reception coming up. You take maybe one more.
Thank you.
Okay. All right. Thank you to the hardest working man in science, Pietro.
So this concludes the symposium today, but we have drinks downstairs, reception. You're all welcome to come and continue to interact.
Also, I would like to thank all the speakers because I think they've done a fantastic job. Can we please give them an applause? Thank you. Thank you so much. And everybody at the Italian Academy who have helped immensely, and Melba and everybody. This has been a great day. I loved it. I hope you enjoyed it too.